that note, thinking about where where things have changed in terms of this topic of gene drives. I think 20 years ago, if at that sort of manor house in where were we? We were in Herefordshire. If you described gene drives, first of all, we would have said that was complete science fiction and it was completely conspiratorial, and don't even talk about that kind of thing. And it's amazing how reality, the external reality, changes as well as the technology over what's not that long a period of time. Through the last 20 years, and looking back to sort of 1997, 1988, we were talking a lot about the problem of genetic pollution, that there was a worry that GM crops would lead to GM traits leaking out into the environment from fields. And what's astonishing about gene drives is it turns that around, and genetic pollution becomes not a problem, but actually the driver of the technology. This is a technology that is based on trying to move traits, genetically engineered traits, out into the wild, out into populations, in fact, to take over and destroy entire populations. So that switch of genetic pollution being a problem to it now being the driver of a technology is kind of interesting. I think if I look back as well 20 years, that was a moment when we were really trying to move of these technologies, of these genetic engineering technologies, into agriculture. And that was what was happening. And now what we're seeing 20 years later is the move of these technologies into conservation, into intentional attempts to change not just agricultural ecosystems, but the wild as a whole. And gene drives are very much emblematic of that. One thing I'd point out is that gene drives are going to actually end up in agriculture. In fact, I think that's really where they're aimed at. But at least the rhetoric is all around changing the wild. It's about changing natural ecosystems. And again, I think if you'd proposed 20 years ago a genetic engineering to change the wild and natural ecosystems, that would have just been thrown out the door. It shows how the conversation has changed, not for the good. I don't know if there's already been some discussion about gene drives in the room.
No, there hasn't.
I'll start by just... There has or there hasn't?
There has not.
There's not. Okay. So for those who maybe are less familiar with what this is, I'll start by just describing the idea of gene drives, which are a particular kind of genetic application, really. And then it comes out of synthetic biology, it comes out of gene editing. I think Michael Antonio was talking about gene editing earlier. And in fact, it comes from before that. There was a theory around that maybe there are certain gene sequences or certain genetic traits which have a kind of almost like selfish genes that are more likely to push from one generation to another. This idea of a kind of selfish gene that will move more quickly from one generation to another and therefore spread in a population. And this idea has been around for about, I think, 20 years. And there was questions about whether these kind of selfish traits, if you could hook something onto them, if you could also hook, say, turning green or something, whether that trait would also spread through a species. But that it was entirely a theoretical discussion. And the idea being that if you could, bias inheritance is the actual term that gets used, if you could make sure that a particular trait will always get passed down to the next generation, then you can change not just the single individual, but you can over time change an entire population. So the normal, when we talked in the last 20 years about genetically engineered traits, let's say herbicide resistance, getting out into wild populations of something like rape and canola and so forth. Really, we were talking there about things outcrossing, but at relatively low rates that might over time become bigger. Because normal Mendelian genetics says that, if you have a trait and you cross the wild species, maybe half the time it's going to get passed on. And then let's say that does get passed on, well then half the time again it might get passed on. So over time, theoretically, that trait will get lost in the wider population. And in fact, this is something that pro-GMO forces, if you like, have constantly said it's all right. This is just going to get, these traits are going to get lost in the wild. Now that may not be true because natural selection, there may be reasons why those traits take off, but all things being equal, that's the case. With a gene drive, the idea is that every time there's a sexual reproduction with a wild species, and you pass on to the next generation, all of the offspring will take that trait. So if as genetically engineered, let's see with the canola example or the rapeseed example, crossbreeds with a wild relative, then if you had a gene drive attached, then all of its offspring would have that herbicide tolerance, let's say, or whatever it is. And then all of its offspring would also do the same, and all of its offspring, and so forth. So you relentlessly push this trait generation to generation to generation, aggressively spreading it through a population until eventually, theoretically, you take over the entire population. So the entire population of brassica, let's say, end up being herbicide tolerant, or if the trait you're trying to spread is that they're going to glow green, they'll all glow green. So the implication being that you release one single genetically engineered organism, but that it's equipped with this gene drive construct. If it works, you can then change the entire population of that organism and related species and so forth. So this is the concept, that you can aggressively spread a gene, an engineered gene trait through a population. Does that make sense?
Mm-hmm. Yeah.
Yes.
And so why would you do that? Why would you want to not just have genetic pollution as a problem, but genetic pollution as the actual center of your technology so that it takes over an entire population? Well, there's a few reasons that you might want to do this. So the reason that's most commonly given is about attacking vector-borne diseases such as malaria or Zika or schistosomiasis. If the trait that you're spreading is one that actually causes sterility, or it's a trait that causes a population to crash and go extinct, then this is a way of getting rid of the mosquitoes that carry malaria, or the mosquitoes that carry Zika. So Anopheles gambiae in this case. So in fact, when we hear about gene drives, mostly what we hear about in terms of public relations is this. If we could drive a gene drive through a mosquito population so that mosquito population ends up sterile, or that mosquito population ends up to be all male, so therefore won't reproduce anymore, then we'll eradicate that mosquito, and along the way, theoretically, we'll eradicate the malaria parasite which it carries. So that's the sort of top poster child for why you might want to drive a trait through a population, in this case to entirely eradicate or change a population. The suggestion there is also that you could do this so that the mosquitoes aren't able to carry the parasite. If you could genetically engineer them so they can't carry the plasmodium, the parasite that has malaria, then you also would eradicate malaria, theoretically. That's not as straightforward as that, but that's the theory. The other reason you might want to do this is around invasive species. That if you have, for example, rats and mice on islands that are eating bird life and taking over islands, if you could release a rat or a rodent that is going to have a gene drive and will turn all other rats and rodents on that island into either sterile rats and rodents or rodents that are all male, these are called dortox mice, then the population of the rats would collapse, and you've removed an invasive species. So that's the other application that we hear a lot about in terms of a wide range of other commercial and even military reasons why you might want to be able to spread a genetically engineered trait through a population. Why, if you look at some of the early patents on gene drives, and I'll get to those in a second, one of the obvious ones is that you can release a weed species so that weed species is susceptible to Roundup or susceptible to a herbicide. So where herbicide tolerance has become a big issue for weeds in the US and elsewhere, something like pigweed, which is now all the way through the US and is a real problem because of the overuse of glyphosate and Roundup resistant crops. and the tolerance has got into the weeds. If you could release a weed species that then became susceptible again to Roundup, then you can keep selling Roundup, selling Monsanto's weed killer. And so you can see why that would be very exciting to Monsanto, who's scratching their head and having trouble with glyphosate resistance. You could theoretically put something into a weed species that would make it susceptible to vinegar, for example. And then, you don't have to use Roundup. You could use vinegar, which would be seen as a benign kind of pesticide, because you've genetically engineered the weed to be susceptible to it. A lot of the work that's going on in agriculture in this gene drive approach is going on with insects. So where you have insects like fruit fly, Drosophila suzuki particularly, that's affecting soft fruit. If you can eradicate those fruit flies, then you can protect your soft fruit. And the same is true actually for rats and mice and voles and so forth. If you can get rid of them, then you're protecting the agriculture supply. It may be an interesting way of speeding up seed multiplication as well, but we can get to that in a minute. And then the last area where you can immediately see this could be powerful is as a bioweapon. And I'll come back to that later. But if you can spread a genetic trait through an area very quickly, then you can not only crash species of rats and mice and mosquitoes, you can also crash the food supply, or in other ways affect the food supply. And potentially, you could also start to target particular populations of people through the microbiome. So we can come to that in a minute. So how does a gene drive work? So the first working gene drive actually was made at the end of 2014. So this is something that was theoretical until just, 3 years ago. 3 and a bit years ago. And the way it basically works is that you use CRISPR, which I think Michael apparently described to you, so that you build the CRISPR system actually into the organism. And so that system, which cuts at a particular place in the genome, and then you can add extra things into the genome, you can actually build into the organism itself that kind of cut and paste. And you can build it in such a way that you also say, cut here and then paste in the trait that you want. And effectively, this is done so that when an organism reproduces, and this is only really works for sexually reproducing organisms, it cuts on both alleles, both copies of the genome that the organism carries with it through each generation, so that both copies of that will have a copy of the CRISPR system and the gene drive trait that you want to drive through. And that then happens generation upon generation. Each generation, you cut into it the specific, cut and paste the trait and the gene drive, the CRISPR system, so that it just keeps going. The first time this was done was by a pair of scientists at the University of San Diego called Ethan Beer and Victor Galantz. They built a gene drive system into fruit flies that turned the fruit flies from brown to yellow. And they basically had a whole big box of brown fruit flies. They put in a gene drive with a yellow trait that would turn them yellow into one fruit fly. They put it in the box. They left it for a few weeks. And then after a few weeks, they opened up the box, and all of the fruit flies that came out of that box were yellow. They said it was like the sun rose in the west rather than the east. It was as if it just totally upturned the rules of genetics, and that's kind of what it is. It upturns the rules of Mendelian genetics. Interestingly, when they did that, they didn't really have a proper biosafety approval from their university. If one of those fruit flies, or two of those fruit flies, had got out into the Californian wilderness, as it were, you could imagine that they would have, over time, bred with other fruit flies, and we would have seen yellow fruit flies spreading across the US. That's kind of the implication here, is that one small change let out into the open ecosystem spreads right across a continent, or spreads potentially globally, if this continues to work. And there are limits to that, we're learning, but that's the theory. At the same time, a scientist called Kevin Esvelt at MIT was also developing pretty much the same technology, and he has a very extensive patent on CRISPR gene drives, which covers their use in agriculture, in conservation, and in other fields as well. And at the same time, a team who have been doing similar stuff at Imperial College started using CRISPR gene drives in mosquitoes. So this team, which is led by a guy called Austin Burt and Andrea Crisanti, are part of a project called Target Malaria. And they receive, I'd say, over $100 million to work on and eventually release gene drives in Africa to counter malaria. Most of that, about $75 million of that, comes from the Bill and Melinda Gates Foundation. And they have a very sophisticated project where they are now in four African countries. They're in Burkina Faso, Uganda, and Mali, but also beginning to do work in Ghana. From the village level right up to the African Union, trying to get acceptance for releasing their gene-drive mosquitoes. Meanwhile, the gene-drive mosquitoes themselves are being built and have been built and are being tested at Imperial College in London, and also a little bit in Italy at Perugia. That is probably the largest single gene-drive project. It's very much presented as a public good because it's about malaria, with the Gates Foundation putting not only the money into Target Malaria, but actually millions of dollars into PR, which we'll talk about in a minute. Probably the second best known gene drive project at the moment is work being done from North Carolina State University and an NGO called Island Conservation. Of species from islands. And so it has what it calls the Daughterless Mouse Project, or GBIRD, the Global Biocontrol of Invasive Rodents Program. And they are working on making gene-drive mice, all of which will be male as they pass on, that they can release onto islands to destroy the mice population on islands. This is a consortium of 7 groups who are working together. There's North Carolina State University and Island Conservation, but there's also the US Department of Agriculture, CSIRO in Australia, the University of Adelaide, Texas A&M University, and Landcare New Zealand, which is a kind of conservation agency in New Zealand. And they're working closely with the US, Australian, and New Zealand governments to identify islands where they want to test their gene-drive mice. They just recently got $6.4 million from the US Defense Advanced Research Projects Agency, which kind of comes to the third major funder here. The first I told you was, so the first major funder is the Gates Foundation. The second major funder is something called the Tata Trusts, who are funding Ethan Beer, who's the guy who originally did the gene drive. And the third is the US military. In fact, we've got some documents coming out on Monday, which you should hopefully see in The Guardian, which show that the US military is now putting over $100 million into gene drive. In fact, is probably the leading developer of gene drives. The US military, particularly DARPA, the Defense Advanced Research Projects Agency, now has its finger in every single gene drive project pretty much on the planet, and is funding most of them, including this so-called conservation approach. And is ostensibly interested in how they would remove gene drives from the environment, given that these could be a very powerful bioweapon. But they're doing that by developing this very powerful bioweapon for supposedly conservation users, supposedly health users. So what's clear is that, in fact, the development of this technology is being structured largely by the military and also by the Gates Foundation. If you look at the timeline, the first working gene drive was probably working at the end of 2014. Within about 4 months, the National Academy of Sciences had commissioned a major study into the implications of gene drives that was paid for by the Gates Foundation and DARPA, the US military. And now what we see is that there is a very close collaboration with the military very much in the middle of the picture, to structure this whole area so that it can move ahead and move quickly to testing. They have a gene drive sponsors and supporters coalition, an advocacy coalition for gene drives. It's going to have a meeting this week at the Wellcome Trust in London, where you guys are. Partly this is brought together by what's been happening politically. That after the national wild, brought the issue, first of all, to the International Union for Conservation of Nature, IUCN, where there was a call for a sort of on biological diversity, where there was a big fight last year in Cancun between the 196 countries of the CBD for a moratorium on gene drives. That wasn't achieved, partly because the funds from the Gates Foundation were used to marshal a lot of scientific support in not stopping research. And now that money, the Gates Foundation has put a further $1.6 million into a private lobbying firm to try and skew the ongoing fight over gene drives. So this is now a very big fight at the United Nations, which is actually going to play out this week. I'm part of an expert group, so is Ricardo Steinbeck, who probably would have been there otherwise. And mostly this week we're going to fight about gene drives because there's such interest from the military, from agribusiness, and from a small clique of industrialized countries. I guess I should just finish with a couple of points. I've been presenting this as a technology which, once you release it, it goes everywhere and it doesn't stop. And obviously that is terrifying. The idea that it will just stop until it takes over a population or crashes a population is an extinction technology. There are kind of two caveats to that. The first is, it turns out probably that gene drives won't be as powerful as they first thought they would be. That, of course, nature and evolution finds a way. That once you release a gene drive organism, it may go some generations, but then there will be resistance, that there will be mutations, and so the gene drive will mutate. Now, it might still continue in some fashion, we don't know, but there'll be a limit to how effective it will be at spreading, but it may be effective enough that it can cause some damage. In fact, Kevin Esfeld, one of the originators of gene drive, recently put out a paper in, I think it was in Nature, saying that he feels now that this should never be used for conservation. He still thinks it should be used for health reasons, but it shouldn't be used in conservation because it's too powerful. The second thing is, people like Kevin Esfeld are now saying, well, we want to take advantage of this. They want to make what they call local gene drives. So the original idea of a gene drive that spread relentlessly until it took over an entire population all over the world, that's a global gene drive. They want to invent locally targeted gene drives that will only go for, let's say, 20 generations, or that will only work in particular subpopulations because they find a particular marker. And that seemed to be the way in which you safely will get to use gene drives. That's also, in some ways, probably the most worrying, because, first of all, they might not work, they may spread. But secondly, this means that you could theoretically commercialize those gene drives. You could imagine gene drives being used as a service for eradicating pests, rather like a sort of genetic pesticide. You also then have a very good targeted bioweapon. If you can say you're going to have a gene drive that's only going to affect certain subparts of the population, it's only going to affect certain parts of a maize population from a particular part, then you have a very powerful bioweapon. So you can see why the US military is extremely interested in this. I think I'll stop there. There might be a couple of minutes for questions. Not quite. Yeah, close but not quite. So that's Kevin Esfeld. And Kevin basically came out and said he was probably wrong to have suggested that they were used in conservation. He still thinks they have a use. I've had lots of conversations with Kevin. I mean, he wishes he hadn't mentioned the use in conservation, but frankly, I don't think it's really his. He's not as powerful as he thinks he is. He still thinks that if you could create local gene drives, then it's okay to use them. And so this is now his argument, is that because he's working on a system he calls the daisy drive, which is a kind of local gene drive that will go a few generations and then stop working. And he thinks if we can perfect daisy drives, or some kind of local gene drives, then it may be okay. But until that exists, he feels they shouldn't be used, which is a step in the right direction, but I don't think you should be developing a daisy drive either. Sure. Yeah. So the UK is one of the key centres for this, of course. The Target Malaria team, who are based at Imperial College, have over $100 million for moving this forward. Some of that money is from DARPA. It's from the Defense Advanced Research Projects Agency. They hadn't disclosed the fact that it was from DARPA. We've just found that out through a Freedom of Information request, and that information, as I say, will be coming out on Monday, so watch The Guardian and other places. Even just that, even the fact that the US military, through this group in London, are funding work to target African communities, should be raising some real questions in Burkina Faso and Ghana and Uganda and Mali. But this it really is, the UK is, because of the Target Malaria Group, and also it seems there's another paper coming out on Monday, which certainly the Science Media Centre is going to be pushing hard. I'm sure you guys all know the Science Media Centre, which is a group at the Roslin Institute who are pushing for gene drives, who are working on gene drives in mammals. So this is it includes gene drives in mice, but also they mention rabbits and hares and other things, mink, for example. So it seems like the Roslin Institute is also kind of stepping into this world. And the paper from the Roslin Institute really does talk about this as being used in agriculture, which is to say not just on islands for removing, it's a conservation, but for actually protecting agriculture, industrial agriculture, through this approach. So I think the UK is becoming quite central. And of course, the third player is Oxitec. So Oxitec, with its genetically modified mosquitoes, isn't using gene drive per se, but is very much involved in this discussion, is very much opening the way to these technologies. Luke Alfie, who is the founder of Oxitec, is right in the middle of this, and I think is one of the recipients of the money from DARPA, from the US military. He's now at the Pirbright Institute. So the UK is absolutely, globally, it's important. And so the places where this is going to be, first of all, I think some real questioning about what this Target Malaria Imperial College funds and work really means is very useful. The role of the UK government in, for example, the Convention on Biological Diversity and other discussions on this, Bioweapons Convention. The UK government, of course, is lockstep behind this. Yeah.
Okay.
Thank you.
I agree. It's also one of the major players. I mean, I'm sure you're having the discussion there about, what's happening more generally on biotech globally, which is this shift to so-called new breeding techniques, synthetic biology. That we're going beyond GM to GMO plus. And the UK is absolutely central to this attempt to get out of any kind of oversight or regulatory process. It's going to be even more central with Brexit, I'm sure. So yeah, I think these discussions coming out in English language and UK publications do travel. Yes, I agree with that. Yeah, and I mean that's even more true now with the mergers, of course. So Monsanto and Bayer are hooking up, Syngenta Chem China are hooking up, and DuPont and Dow. So we're really down to 3 or 4 players now, and that's just in seeds and inputs. I think the bigger picture is what's happening around data conglomeration. That a company like Monsanto or DuPont now considers itself to be a data company, and we're moving into this era of precision agriculture, where basically it's about data driving driving agriculture, but also driving biotech and so forth. And so actually what we're going to see is the big players are whoever can accumulate and control data through artificial intelligence algorithms, and then the machine companies. It's going to be John Deere. It's going to be Case New Holland. We're seeing right now a major reconfiguration of who controls this technology, and it's all about big data. So I think that's right, and I think it's also important to look at what's happening now, not where we were 5 or 6 years ago, because it's really shifting quickly. And of course, one of the things the gene drive work and this sort of attempt to engineer the wild is showing us is we have to not lose our eye on the military. I think in the fights over GMO crops, we focus very much on agribusiness and seed and chemical and input companies, and that was appropriate. And we kind of took our eye off of the role of militaries because they weren't so, in the end, weren't so involved in that technology. That's not true now. The largest funder of synthetic biology development, so not just gene drives, but far beyond gene drives, is DARPA, the Defense Advanced Research Projects Agency in the US. It's the largest public funder anywhere in the world. And they have, quite aside from the work on gene drives, they have equally distressing, as far as I'm concerned, projects about using insects to deliver vaccines and viruses to plants. They have projects around trying to engineer what are called ecological niches. They've got a whole bunch of really quite concerning stuff, with in each case tens of millions or hundreds of millions of dollars behind it. And so we need to also keep the military visible in how biotechnology as a platform is now being put together.